Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning sessions in Biotechnology Practicals. Here in this semester, you will be learning about the practicals of immunology and medical biotechnology. In immunology, few experiments are involved like blood typing, Octraloni double diffusion and Widal test, etc. And in this session, we learn about how blood typing is done. First and foremost, we should know what is blood typing. Blood typing is the technique of identifying the blood group antigens, or on the surface of the blood cells we will find the antigens, and these antigens will be specific in an organism, and the technique of identifying the type of the antigen present on the RBC is called as blood typing. And this technique was first discovered by the Karl Landsteiner in the year 1900. Let's see how we will do the blood typing. Take a clean glass slide, and then draw three circles. Draw three circles and label them as A, B and D. Then unpack the monoclonal antibody kit. So these antibodies will be raised against antigen A and antigen B. Along with that, we also have the antibodies which are prepared against the Rh antigen. In your theory classes, you all would have learnt about the blood group antigens. The RBCs on their surface, they have either antigen A or antigen B. And in addition to that, few cells also have the antigen that is sharing the similarity with the rhesus monkey, the proteinaceous substance that is present on the RBC. So that is called as Rh antigen. And there are certain RBCs where they have both AB antigens. They have both AB antigen. And few RBCs will not have both A or B, or they will not have any antigen on their surface. So based on the presence of the antigen present on the surface of the RBCs, blood groups will be classified. And here these antibodies are monoclonal antibodies which are raised against the specific antigen. For example, anti-A monoclonal antibodies are raised against antigen A, and anti-B antibodies are raised against antigen B. And you have the anti-D anti-sera which is raised against the Rh antigen. So these synthetic antibodies will be recognizing or identifying this specific antigen in the blood group or the blood serum. So you take a clean glass slide, draw three circles and label them A, B and D. A refers the identify the A antigen, B identifies the B antigen and D identifies the Rh antigen. So when you put the blood droplets here, so they will react with the respective antisera and you will come to know which is the blood group in the given blood sample. So take out the kit containing the synthetic antibodies, keep them on your working table. Then add anti-A antisera to circle labeled A, anti-B antisera to circle labeled B and add anti-D antisera circle labeled D. So you are adding only one drop of the solution respectively to their circles. After that, so you the person whose blood group has to be identified, you call him or you take your blood sample. How you will be taking the blood sample? So now wipe your middle finger or the ring finger with the alcohol swabs. So you are wiping the surface of the finger with the alcohol swab. Take the cotton, dip it in the 70% alcohol and then wipe the surface of the fingers to avoid the contamination or to remove the any microorganisms or anything that is present on the surface of your finger. After that, rub gently near the fingertip. So push all your blood or rub towards the tip of the finger, and using lancets will be taken. Lancets or uh, the needles are taken, and these lancets will be fresh. So you have to open these lancets when you are pricking your finger for the blood. So the sterile lancets will be taken and prick the tip of the finger with the end of the lancets. You should not touch the tip of the lancet, otherwise it will be contaminated. And then pour or put drop of the blood into each circle. So here what you are doing, you are swabbing your finger with the alcohol containing cotton, then air dry it, then you are taking the sterile lancets and then you are making a puncture, you are puncturing the finger, and you are taking, or your blood will be taken out and it will be dropped into the circles. Now, after adding the drop, so add a drop of blood to each circle without any contamination or the mixing. So you should add the blood drop without touching the solutions of A antisera or the B antisera or the D antisera. So just above the circle you drop the blood, one drop blood will be added. After that, after adding the blood, one drop to the respective circles labeled as A, B and D, with the fresh toothpick, separate toothpick, you have to mix the solution. You have to mix the solution. The toothpick which is used here in the A circle should not be used in the B. So separate toothpick you have to take, and then you have to mix the blood immediately with the antisera solution with a separate toothpick, and there should not be any contamination. After that, you observe for agglutination. What is agglutination? Here we have the antisera, and in our blood we have the antigens. We have the antigens. So when you add this antigen in the blood into the antisera solution in the respective circle, there will be interaction between the antigen and the antibody, or there will be interaction with the blood with the specific antisera that is loaded in the circle. If the person has the specific antigen, for example A antigen on the surface of the RBC, there will be clumping of the blood, or the agglutination of the blood takes place, and that results in the separation of the liquid from the precipitated or agglutinated blood cells. So that is called hemagglutination, or the clumping of the blood takes place. Now, if the person is having the blood group A and Rh antigen on the surface of the RBC, then he will show the blood clumping, or the blood clotting will be observed in the A circle and the D circle, because A blood group will be having the antigen A and he will be having the antibodies for the B. Anti-B antisera will be there in the blood, and hence so the A blood group individual show the clumping in the circle A and D, because he is carrying the Rh antigen on the surface of the RBCs. So that's why we'll find the precipitation both in the A circle and the D circle. So he is having the A antigen and anti-B antisera. So this anti-B antisera will not interact with the A antisera. So it is only the antigen which is interacting with the anti-A antisera. So such type of individuals are called as A positive. The positive refers to the Rh antigen, refers to the presence of the Rh antigen. So if the blood group containing A and the Rh antigen on the RBC, he will show the precipitation in the A circle as well as in D circle. So if the person is having the blood group A but without Rh antigen, he doesn't carry the rhesus monkey antigen on the surface of the blood cells. Then how the clumping of the blood takes place in the blood typing. Here we will find the precipitation in case of the circle labeled A, but not in D. So he is considered as A negative blood group. His blood group will be considered as A negative because his blood is showing clumping only with the agglutination with the anti-A anti-sera, but not with the anti-D anti-sera. So that's why it is called, this blood group is referred as A negative blood group. Next, if the person is having the blood group B with Rh antigen on the surface of the RBC, then he will show the blood clumping in the circle B and D, because the blood group B will be having the B antigen and he will be having the anti-A anti-sera in his blood. Anti-A anti-sera in his blood. That's why his blood shows the blood clumping or the blood cell clumping or the agglutination in case of the circle B and in the circle D, because he carries the Rh antigen. That's why he is considered as B positive. He is considered as B positive. So, you have the B antigen on the surface of the RBC and anti-A anti-sera in the serum. So, this B antigen is interacting with anti-B antibodies that is loaded in the circle labeled B. Now, if the person blood group is B without Rh antigen, so the person blood group is B without Rh antigen. Now, his blood shows the clumping in the circle labeled B and his D circle will not show the clumping. The blood will not interact with the anti-D anti-sera. That's why clumping will be absent in the D as well as A, because A antigen is not there on the surface of the B RBCs. Only B antigen is present on the RBCs. So that is interacting with the anti-A anti-sera. Since there is no antigen on the surface of the RBCs, that will not interact with the anti-A anti-sera, and hence the Rh antigen will not interact with the D anti-sera and A antigen will not interact with D anti-sera. But he carries the B antigen that will interact with the anti-B anti-sera. So, such type of individuals are called as B negative blood group containing individual, and he does not carry A antigen and also Rh antigen. So, that is regarding B negative blood group containing individual. They have B antigen and anti-A anti-sera in their blood serum. Now, suppose the individual has the blood group AB on the blood group AB, or if the RBCs carry both A antigen and the B antigen on the surface of the RBCs, along with that he also has the Rh antigen on the surface of the RBCs. In such condition, you will find the clumping in A circle, B circle as well as in D circle. So, you have the agglutination in the circle labeled A and circle labeled B and circle labeled D, because these RBCs on their surface they have AB and also Rh antigen, due to which the A antigen will interact with the anti-sera in the circle containing anti-A anti-sera, and B antigen will interact with the circle which is having anti-B anti-sera, and Rh antigen will interact with the anti-D anti-sera which will bind to the Rh antigen. That's why individual with the AB blood group shows the agglutination in all the circles labeled A, B and D circles respectively. They interact with the A antigen and B antigen, and the anti-Rh antigen will interact with their anti-A anti-sera, anti-B anti-sera and then anti-D anti-sera. That's why you will find the clumping in all the three circles. Next is, if the person on the RBC surface, he has both A antigen and B antigen, but he is not carrying the RH antigen. In that condition, what will happen? When the blood is dropped into the A circle, B and D circle, we will find the agglutination in the A circle and in B circle, but not in the D circle, because even though we added the anti-D antisera here, there is no RH antigen on the surface of the RBC to interact with the anti-A antisera. That's why you won't find the clumping or the agglutination in the anti-D antisera or in the circle labeled D. That's why the individual will be considered as the AB positive. AB positive. So this is regarding the identifying the blood group of an organism, either by presence or absence of the agglutination will decide the blood group of the individual. Next, suppose if the RBC is not carrying any antigen, A or B. A and B are absent, but he has only RH antigen on the surface of the RBC. Then in such condition, when you drop the blood of such individual, you will not find the clumping in the circle A, and you will not find the clumping in circle B, and you will find only hemagglutination in circle D, because this RH antigen is interacting with the anti-D antisera. Though these individuals will not carry A and B antigens, but they carry anti-A antisera, anti-A and anti-B. But these antisera will not interact with our antisera. It's only the antigen is interacting with the anti-A antisera, synthetic antisera. So these individuals, they will not carry the antigen A and B, but they carry antibodies, that is A and B. They carry both A and B antisera, but they carry the RH antigen. Antigen A and B are absent, but they are positive for the RH antigen, due to which such individual who does not carry the A and B antigen on the RBC, but they carry the RH antigen on the RBC, they are called as O positive. They are called as O positive. Okay, this is regarding O positive blood group identification. Next is, if the person is not having the antigen A and B, but he carries anti-A antisera and anti-B antisera in his blood serum, but in addition he is not carrying the RH antigen also. On the surface of the RBC, you don't have A, you don't have B antigen. In addition, the RBC is also not carrying RH antigen. But in the blood sera, you will find anti-A antisera, anti-B antisera, and this individual will not show the hemagglutination in circle labeled A or B or D. Such individuals are considered as O negative. Their blood group is considered as O negative. So, here the individual on the surface of the RBC, the person is not carrying the antigen A, B and RH, but his body has the antibodies for all the missing antigens. But the surface of the RBCs are not carrying any antigen, due to which when we do the blood typing on the slide, Karl-Landsteiner blood typing, you will not find the clumping in any of the circles having anti-A antisera, anti-B antisera or anti-D antisera. So such type of blood group is called as O negative blood group. So now you came to know about A positive negative, and then B positive and negative. Next, you learnt about how to identify the blood group AB positive and negative. At last, you learned how to identify O positive and negative. So, this is regarding identifying the blood group of the given blood sample using the synthetic antibodies that is developed by the Karl Landsteiner. So, this blood typing will categorize the individual into 8 blood groups like A positive, A negative, 2. Then B positive and B negative. So, here 2 types of blood group, here 2 types of blood group. AB, again you will be having one group will be AB positive and another will be AB negative. Similarly, you have O positive and O negative. So, in each type you have the two groups of blood grouping, blood types, and finally, so the whole human being blood group can be classified into 8 major groups, that is A positive, A negative, B positive, B negative, AB positive, AB negative, and finally O positive and O negative. All this principle of this experiment depends on presence of the antigen and the presence of the antibody for the missing antigen. So, this principle will be used. So, antibodies which are present in our body will not interact with the Landsteiner blood, antiserum monoclonal synthetic antibodies, but it is only the antigen is interacting with the antisera that is loaded in the circle A, B and D. So, this is regarding how to identify, determine the blood type or the blood group of the given blood sample using the Karl Landsteiner. When you do the blood typing in the laboratory, you will find the clumping of the blood like this. So, this precipitated the blood cells get separated from the liquid. For example, here, so here you are finding the clumping in the A circle and the D circle. So, this is A positive. So, here you can see the blood. So, this is the blood sample without any antisera. When it is added with the anti-A antisera, so since there is antigen on the RBC is interacting with the anti-A antisera, that cells get cellular portion gets separated from the blood, leaving behind the liquid. But here in anti-B antisera, there is no clumping of the blood cells because there is no B antigen. In anti-D or the circle labeled D, you will find the clumping of the blood cell because of the presence of the Rh antigen. So, that is why you will find the clumping in A circle and the D circle. In case of this is regarding B positive. So, here in the B positive blood group, in A circle you won't find any clumping of the blood. So, the blood is similar to the untreated the blood sample, but in case of the circle B with anti-B antisera, it is interacting and it is giving the clump, and in the D antisera, the D circle also it is interacting with the anti-D antisera. So, that is why you will find the clumping of the blood in the B circle and the D circle. And in case of AB positive blood group, AB positive blood group, you have the clumping in A circle, B circle and the D circle, because the blood cells have the A antigen, they have B antigen on the surface of the RBC, and then in addition they also have Rh antigen, due to which you will find the clumping of the blood in all the 3 circles. But here you can see the little clumping of the blood is observed in the last one, where A is not having the clumping, B is not having the clumping, but here you will find the clumping in very low concentration. So, this is O positive. This is O positive, where D circle is showing the clumping. It is showing the clumping. So, this is regarding the observation or the appearance of the slides under laboratory condition when you do the blood typing with the fresh blood sample and the Landsteiner blood typing kit. So you should remember this to identify the given blood group. Where you will find the agglutination, so that indicates the presence of the particular antigen. Absence of the agglutination, absence of the antigen. So this is regarding blood typing. Here you will find the antigen in A, so it is A positive, antigen in B, so it is B positive, antigen both in A and B, so it is AB positive, and only in D then it is O positive. And based on the presence or absence of the antigen, the blood group can be classified as universal acceptor or universal donor. So AB positive is universal acceptor where it can accept the blood from the A and from the B and also from the O positive. And this O positive is considered as universal donor. Universal donor. Donor because this blood group could be used to the individual having AB, could be used to the individual having B, could be used to the individual having the blood group A. But the person who is having the O blood group should receive the blood only from the O individual. That's why he is considered as universal donor. Whereas AB positive is considered as the universal acceptor because he has the A antigen, he has the B antigen and the Rh antigen. So these will not have the problem with the A blood group or the B blood group and even the O blood group. O blood group is not at all carrying any antigen. So he can accept the blood from any of the type of the blood group containing individual. Such blood group containing individual are called as universal acceptors and O blood group containing individuals are considered as universal donors. So this is regarding blood typing. I hope you understood. So take a clean glass slide and then take the blood typing kit, open and keep it, and then you label your slides into 3 circles, label them A, B, C, A, B, D, and to the respective circle, A circle you add anti-A anti-sera, to B circle you add anti-B anti-sera, and to D circle you add the Rh antibodies, that is designated as anti-D anti-sera. After that you have to drop or pour the blood, one drop of blood you put it on the surface of the circle without mixing, and then you mix the blood with the anti-sera with the separate needle. Each circle should have the separate needle. Mix and observe for agglutination. Presence or absence of the agglutination will decide the type of the blood. Okay, so in this session we learned how to do the blood typing under laboratory condition. I hope you understood this experiment. Thank you.